Hello dear friends, welcome to Homeopathic Lighter. Topic is microbiology whole syllabus. We will discuss in this video question and answer. What are the most important questions are asked mostly in your university exam and BHMS or competitive exam? We will cover in this one video. So without further late, we will start our discussion. First, this is the paper in which I write all those questions which are important and mostly oftenly coming in every year in your exam and also in your vivas, also in your upcoming university competitive exam. You can see that is KLB, those things. What is KLB? You know, KLB, diphtheria. It is coming from diphtheria. So KLB we will discuss. Describe the morphology, cultural characteristics and lab diagnosis of diphtheria. First question we will discuss. So KLB is full name is Corynebacterium diphtheri. Okay, Corynebacterium diphtheri. Why it is called as KLB? Here is the answer. KLB or Loflor bacillus. KLB and Loflor. These are two scientists who firstly discovered this diphtheria bacteria. That's why as their name, its name as KLB or Loflor bacillus because it is coming from bacillus group of bacteria, gram bacteria. They are gram positive bacillus. Okay, and they do not contain any spore and they are not motile. They contain metachromatic granule, which is what is metachromatic granule? Metachromatic granules which gives the energy to the bacteria. That's why it is contain the polymetalphosphate granule. Then what is the clinical feature? Mainly it produces pseudomembrane by causing airway obstruction. So it causes airway obstruction. Its main action is on the respiratory system. So it is systematically causing myocarditis and polyneuritis. What it causes? It causes myocarditis and polyneuritis. What is myocarditis? Myocarditis is the cardiac inflammation and polyneuritis means many nervous organs, multiple nervous system nerves are affected due to this bacterial infection. Then what is the pathogenesis? Pathogenesis is that when this bacteria when enters through the nasopharynx, it causes through the respiratory droplet. So from this line, what we conclude that the mode of transmission is through respiratory droplet, causing grey fibrous exudate, and which grey fibrous exudate forms in the respiratory tract, which is composed of bacteria, WBC, necrotic mucosa, okay, which causes the block of airway. So first thing it causes by transmission, it causes the block of airway, and this block of airway occurs by the secretion of a diphtheria toxin. So diphtheria is mostly toxic because of its toxin. So what the toxin contain? The AB toxin, which is inactive and which causes what it causes? This toxin causes the our DNA or our cell function damage by causing inactivation of elongation factor, prevent the protein synthesis of all the cells which it is infected. So causes arrhythmia, myocarditis, nervous peripheral neuritis, cranial nerve palsy. These are things occur. So we can know that how the toxin is how the toxin is acts in our body by lysogenic pathway. So lysogenic pathway through which the toxin is released to the means if we take this is a cell, so diphtheria when act its toxin is enters to the cell by the lysogenic pathway. Okay, this thing is called and which causes systemic disease, means disease related to blood vascular system. So here due to this what happened, there is in the throat there is a bull neck appearance is found. Bull neck appearance, you should remember which bacteria causes bull neck appearance, that is a diphtheria bacteria. So what is bull neck appearance? That is the presence production of the pseudomembrane in the tonsillar and the larynx area that causes bull neck appearance. Important question mostly asked, which bacteria causes bull neck appearance. Now the second part we discuss, that is our lab diagnosis. How we will diagnose the diphtheria bacteria presence? By taking the throat swab. So by taking the throat swab, what we first think, we divided into two parts. One is we use for the throat swab for gram staining and a culture staining. Culture is the most sensitive one because by culturing we can know whether it is a staphylococcus or a streptococcus or it is a diphtheria. So by potassium tellurate and the LSS media we use for the diagnosis, that is Lupus serum slope media. Then now the other media which is used Holles media, macular media, translated media, and important is that this media, when we culture, we see the uniform arrangement, means Chinese like arrangement we will find. And one sick test is also sick test, it is also commonly asked in your short note. What is sick test? So sick test is usually done for the susceptibility of the person towards the diphtheria. So how much the person is susceptible to this we can know by taking the test of a sick test. Now we go towards the we can know that blood culture no use for diphtheria. Why? Because there is no you can in the blood culture this culture is not fine, because diphtheria infection is due to the toxin, not due to the diphtheria bacteria. That's why we should remember that the blood culture is negative for this diphtheria. It is an important mark for that diphtheria. Then diphtheria toxin is made up of two components. We discuss A and B. What is A? ADP, ribocycling CFF, and the B function is to binding that toxin A to that cell, helping the binding of toxin A to the cell. So every colony bacteria is not pathogenic. Why? Because we discussed before that the tox gene is important for the infection and for the pathogenesis inside the body. So every colony bacteria is not the pathogenic, but its pathogenic effect is due to the toxin. So this toxin is having the lysogenic conversion, and which causes the toxin by lysogenic conversion and transduction. The bacteriophage method, it enters to the cell and causing cell DNA levels alternation. Okay, so with this we have the cutaneous diphtheria lab diagnosis, through which we puncture the ulcerated region to find the toxin, and the treatment is the erythromycin, penicillin, and if the protective titer also done for the further analysis of if diphtheria occur or not, or vaccine is also now available. So with this, we first question our Leishmania, our KLB we discuss. So in KLB we will discuss all the lab diagnosis and pathogenesis of KLB. Okay, now we go towards the what Leishmania Donovan life cycle and lab diagnosis. So here is the life cycle of the Leishmania Donovan. So in the human, one is sandfly. So what human cause? In human there is indefinite host. Human is the definite host, and sandfly is the indefinite host. Why? Because there is the sexual reproduction occurs in the sandfly region. That's why we discussed first that when the sandfly from his midgut, it is released the promastigot form, which is the infected form, which is released to the human. So that what happened, the metacyclic promastigated invade the human body, then enter first. It is mainly sensitive to the macrophage, granulocyte, and some. But these macrophage and mononuclear cell, they are having the tendency to destroy the promasticated. So many promasticated form, from which only some will invade and causes destruction, because granulocytes having more tendency to causes killing of the this promasticate. Okay, that is the reason why there is more incubation period is in the is 2 to 3 weeks because of this infection. That's why they are having more amastigate requirement because of the infection. To clinical symptom to find, we have to amastigate form should be rise, and this occur by the simple division. When the cell enters the promasticated, when enters to this macrophage, then there is in the cell there is division of the promasticated occur, and it is transferred into amastigate form. And when amastigate is released from this cell, we know that amasticated, when many amasticated is lodges inside this cell, they burst out and they comes out outside, and this amasticated is having the resistant wall, so that the granulocyte or phagocyte macrophage they can't they cannot kill this amasticated form. So some amasticated again enters to the other phase cells and causing more and more infection, and some are enters to the some are enters to the outside, and this sandfly when infected by this when the sandfly reach to that transmitted zone, that is a stool full, any stool or urine or any infected material, when sandfly sit, the amasticated enters to the sandfly and release in the midgut, and this midgut region amasticated are released and transported into procyclic promasticate. So then promasticated becomes converted into metacyclic by simple division, and then migrate to the pharyngeal part of the sandfly, and it is by the feeding of the during the meal period of the sandfly, when this feeding of this promasticated enters to the mammals, and it forms as a metacyclic promasticated occur. So in this way the lab diagnosis of the life cycle of the leishmania we discussed. Then what is the lab diagnosis? There is two things. There is direct evidence, another is indirect evidence. In direct evidence, there is we do amasticated staining smear by taking the blood smear or bone marrow aspiration or a splenic aspirate. Why? We know that amasticated form is the infective form. The clinical feature which are coming from this leishmania due to this amasticated form. So that we should demonstrate this infective form by taking the blood or a bone marrow aspiration or a splenic picture, because promasticated cannot remain in the blood because they are killed by the macrophages. Because amasticated resistance to the macrophage, that's why they are identified by the peripheral blood, bone marrow, and a splenic aspiration. Then indirectly it is serodiagnosis by ELISA technique and by PCR technique, and the specific leishmania test is also now coming into the lab for so that we can, by skin test we can know whether it is a leishmanian or not. Now we go to the varicella zoster virus, which is coming as a question, that is pathogenesis, etiology and complication of a varicella virus. So etiology is that varicella virus contain causing two diseases, that is chickenpox and zoster. Okay, if you like, then thumbs up our video, so that we will encourage and more will this type of a video we will provide more and more. But so like, subscribe to our channel. So without further delay, varicella zoster virus having two types of a disease, that is chickenpox and a zoster shingles. So chickenpox causes what? It is a contagious disease, mainly affect children, but they are not fatal in the children. But the adult and those person who are immunocompromised, they are having the more chance of infection, because of the fatality is more in this person, in this location. So it causes generalized vesicular eruption. But in the zoster or shingles, there is what happened? Oh, sorry. In this zoster, what happened? There is a vesicular eruption occur, and at the distant part. Okay, at the distant part there is a distinct, those part where it is infection occur, this part is only affected, not generalized infection is not found, and its main action in the trigeminal ganglia, and the vesicular access mainly occurs in the trunk, head, neck, and it causes trigeminal neuralgia. So what is the pathogenesis? It mainly causes respiratory skin and a portal spread by the upper respiratory tract and conjunctiva, by lymph node, blood, liver, spleen, spread by skin. It causes the ballooning degeneration, causing accumulation of a tissue fluid in the vesicles, such as in the chickenpox. What happened we see. Then respiratory causes respiratory secretion increases by action on the mucous membrane, so that it is more easily transferred to one infection to other. It is easily occurs by this respiratory route and a neural degeneration. So in this way we will discuss the pathogenesis. Another is the complication, as its main action in the CNS and the nerve system. So CNS neuralgia, or in pregnancy, mother and baby will affected, because this cross the placental barrier, and a pneumonia will be found in the zoster case. And the Rice syndrome, which is the important syndrome which is only found in this varicella zoster virus infection. Now our next question. Next question is about the influenza virus. So what is the question? That is morphology of influenza virus, lab diagnosis, and pathogenicity and the antigenic property of the influenza virus. Now what is the morphology of influenza virus? You can see in this view that is influenza virus. Here you can see that it has four component we should remember. One is it is content of a nucleoprotein, that is an RNA surrounding lipid envelope, and there is a spike-like structure which is comes two color. One is blue, another is red, and red is contain RBC, and blue is the neuraminidase. That is a protein which helps in the invention of the influenza inside the body. Then capsid. This is all about the picture view. So it is made spherical. You can see diameter is 80 to 120 nanometer. Pleomorphism means it can change its shape. Many morphological structure it can vary. The core contain RNA protein, that is RNA nucleoprotein. Now mainly influenza have two types of antigenic property, that is antigenic drift and antigenic shift. What is antigenic drift? Mainly there are two types of virus of type of influenza virus which infect the living organism, that is virus A and virus B. Virus A, here you can see if the virus A is converted to virus B by the mutation of any one spike, that is in hematoglobin mutation if occurred, then it is converted to virus B. Okay, but if virus A and virus B both are mixed together and form another type of a virus, that is virus C, then we can say that is antigenic shift. So what is antigenic shift? Antigenic shift is the intermixing of a virus A and virus B by the changing of the both nucleoamidase and the hematoglobin forms a virus C, which is another type of morphology, and that's called as antigenic shift. But in antigenic drift, only one component undergo shifting, that is only one component when shift, that is called antigenic drift, that is virus A converted to virus C, only mutation of the hematoglobin level, hematoglobin glutamine level, that is called antigenic shift of the antigenic property of the influenza virus. Now we go towards the pathogenicity of the influenza. Now the cultural characteristics of influenza. So mainly it can grow in amniotic fluid and a chick embryo. So how can we identify the influenza virus? If the minimum amount of influenza virus appears in the blood or any culture then we go for the cultural determination, and by culture we give that influenza virus in amniotic fluid and the chick. It can also grow in monkey kidney cells in the influenza virus, and it is inactivated at 50 degrees centigrade for 30 minutes. If we and it is also inactivated by the ether and formaldehyde, phenol and ether. So these are resistance to this medium. Now the antigenic we discussed, then pathogenicity. So influenza virus mainly the route it enters into the body, that is a respiratory route. The function of a neuraminidase, which we see in the morphology, a spike structure which helps in the reduction of the viscosity of the mucus and promote the virus absorption on the cell surface. You can understand if this is the passage. Here the mucous membrane is lined, and if the influenza virus comes, the spike-like structure, that is neuraminidase, it causes what is epithelial surface and promotes to the entering of the virus into the blood surface. So this is all about the function of a neuraminidase. Now that's why what happened, the cell which is trachea or bronchi, the cells of they are also got remains damaged, and the incubation period is 1 to 3 days. And the clinical manifestation, what are the signs? That is fever, headache, generalized myalgia, respiratory symptoms all are found. Without complication it resolves in 2 to 7 days. But if the complication arise, then medicine should be recommended. That is pneumonia due to the bacterial super infection, means more than encephalopathies and a Reye syndrome. These are caused by the complication of influenza virus. So this is all about the influenza virus. Now we go to the question which comes as a streptococci classification, and what are the classification of streptococci, and what is the pathogenicity of the streptococci in the sore throat lab diagnosis. This is the point which we discuss now. That is streptococci, it is based on the presence or absence of oxygen. It is one is obligate anaerobes and there is facultative anaerobes, and live easily, but obligate it is anaerobes, like they do not require oxygen. Oxygen not required in anaerobes. Okay, oxygen not required. Here oxygen should require. Here presence or absence of oxygen it can okay to that streptococci. Okay, now we go for the classification, that is in the aerobes, that is alpha types. Alpha is the viridian, where alpha and beta is occurs in the basis of hemolysis. So if the hemolysis occurs, it is beta. So if not, then alpha, there is physiological and biochemical properties. According to this, the alpha is also divided. In beta, there is hemolytic streptococci because there is group A streptococcus, and the classification is occurs in the A, B, C, D up to P. Okay, and the gamma is the species groups under enterococcus. Also, it is also divided, and the beta is important because it causes most of the infection, beta hemolyticus. And if the serological typing occurs of beta by M protein or Griffith typing. Now, what is the pathogenicity of the Streptococcus aureus? So, streptococcal pyrogen, it is the important streptococcus which always causes the respiratory infection. So, its primary site is the throat. That's why sore throat is the most common tonsillitis of the pharynx. More diffusely causes pharyngitis. So, virulent group of A group of streptococci adhere to the pharyngeal epithelium. So, when adhere, they enter to the by means of a pili, and the glycoprotein fibronectin on the epithelial cells probably serve as a lipoic acid ligand. So, through this ligand, the streptococci enters to the invade the epithelium of a tonsil or a throat. So, tonsillitis is more common in the older children. Why? Because who commonly develop diffuse tonsillitis. Localization occurs on the basis of a hypersensitivity reaction. So, in throat, there is a streptococci spread to the surrounding tissue, which leads to also complications like suppurative otitis media, mastitis, Ludwig's angina. Okay, but here the meningitis never occur, it is rare. Streptococcal pneumonia also follows to this streptococcal pyrogen. Followed by streptococcal pyrogen, streptococcal pneumonia can occur, and which leads to the pneumonia. So, this is the pathogenesis of a streptococcal sore throat. Now, lab diagnosis by culture, by swab, but here the smear having in the throat infection, there is a smear of a throat. If you take the tissue and cause smearing, then we can't find invade the and remain as a flora of bacteria as a like of adaptation inside the flora. And by fluorescent method, we can distinguish the gram A streptococcus, group A streptococcus, and there is also a streptococcal kit now available, so that within a 10 to 15 minutes we can diagnosis occur easily. And the most sensitive test is the streptoenzyme, that is very much sensitive and very much nowadays applicable in the streptococcal sore throat lab diagnosis. Now our discussion is Toxoplasma gondii, morphology, then life cycle, lab diagnosis, pathogenesis of Toxoplasma gondii we will now discuss. So, here you can see this is the picture of a Toxoplasma gondii. Okay, this is the picture of a Toxoplasma gondii. You can see there is a Toxoplasma gondii is a protozoan parasite, okay, and it is present inside the cell, so it is an intracellular parasite. So, mainly Toxoplasma gondii is the which type of parasite, that is a protozoan we discussed. Then it contains one cell, that is one celled eukaryote, infects warm-blooded animal. Mainly warm-blooded animals are infected. This is the you can see this is the structure of a Toxoplasma gondii. It contains the endoplasmic reticulum, then our inner membrane, another one is outer membrane. With this there is a microsome, and in the tail position there is a tonoid and the rothotrite are present, and also microtubule and mitochondria is present because it is eukaryote. Now we go plus the morphology. So, Toxoplasma gondii it's a two form, one is a asexual form, another is a sexual form. So, asexual form is mainly consist of a tissue cyst which is formed from the tracheozoids. So, sexual form is a oocyst. So, oocyst converted into tracheozoid, converted into tissue cyst. Tissue cyst is the body joist. Now, we discuss one by one the stages. Tracheozoid is a crescent in shape, infect all the nucleated cell except RBC. So, inside the whole cell, what it causes? It causes parastreponemus vacuole formation by dividing asexually and form the internal body. So, this is the picture of the tracheozoids. Now, we go to tissue cyst. Tissue cyst is the resting stage where the chronic infection is start. So, trophozoid with presence of interferon, gamma interferon, heat, shock protein, and pH with optimum pH and temperature, it is converted into bradyzoids. Most common tissue cyst is formed in the muscle and the brain. Okay, oocyst is the sexual form we discussed, and it is the host is the definite host is the cats. Now definite host is cat because the sexual form is occurs in this cat region, and the intermediate host is man. Man, goat, sheep, pigs, many others are found. Now we go towards the life cycle. So first we see that in the man, that is rodent or mammals, they got infection by the injection of a food and vacuole which is infected by the oocyst of the cat. If the cat gives its oocyst form to this milk or any contaminated food, then it enters to the man through mouth. Then inside the mat or mammals, they got accessory egg system inside the intestine and form the sporozoids or bradyzoids. Then these bradyzoids or sporozoids invade the epithelial tissue and forms the tracheozoid. And the tracheozoid undergo endodyne and spread metastasis occur and spreads to the lymph nodes. Okay, so this cycle occurs in the human cycle or intermediate host cycle. So there is what happened in the human cycle. There is the spreading occur by these tracheozoids. So then inside the extraintestinal organ, there is a transformation of tracheozoids into bradyzoids, and then tissue cyst is formed. Either the tissue cyst directly enters to this another normal healthy cells to infect more and more, or it enters to the cat by infection. Contaminated cyst enters to the cat, and inside the cat the cycle which is starts, that is a sexual cycle. So bradyzoids release from the cat's intestine, enters into the form schizogony, and this schizogony undergo bradyzoids to form male and female gametogeny. Then fusion occur and zygote is formation occur, and this zygote is in the further development forms the sporulate oocyst, and which is now the infective stage and which causes the infection. So this is the infection part, or the life cycle we discussed. In pathogenesis, mainly the geographical area, cold area, there is more chance of occurrence than warm area. Age is older age of the fetus. Exposure to cat or cat fecal or uncooked meat, if eat, then these things happen. Then toxoplasmosis is a immunocompetent disease. Both humoral and cellular immunocomplex is affected by this toxoplasmosis disease if the infection occurs. Acute stage, it is asymptomatic. In the chronic, mainly causes cervical lymphadenopathy, occipital, supracervical, inguinal lymph node is main action, because we discussed in the life cycle the tracheozoids are invaded by the metastasis into the lymph nodes. That's why they are mainly action, mainly causes lymphadenopathy. Toxoplasmosis is occur in immunocompetent just like HIV or any type of DIC, or causes inflammatory necrosis and the inflammatory response it has. So that in this way the toxoplasm type cycle, morphology and the pathogenesis we discuss.